Yeah, so I guess I failed to mention what the purpose of the panel was, which was to discuss the future of CRISPRs in therapeutic applications. And I guess you can just gather from the introductions that the panel is a little bit unbalanced in that regard with respect to expertise. You have 3 scientists who work in pharmaceutical companies who work in discovery, and then only 1 expert in the CRISPR therapeutic field. So with that in mind, we'll try not to gang up on Deb too much, but I guess we will be looking at her to give us the on-the-ground perspective to a lot of the issues and questions that we've been kind of discussing online as we prepare for this panel discussion. So let me lay those out, and also I'll encourage, if the audience wants to ask some questions, I think we'll be able to interlace some of those as well. But what I'd like to do is kind of pose 3 major areas to the panel, get their impressions and their viewpoints on that, and then if we have some time, like I said, we can have the audience add some questions as well. So in kind of just discussing this through email, I think we all came to a basic conclusion that a lot of the issues that are facing CRISPR therapeutics are no different than any gene therapy platform right now that's trying to get their products into the clinic. And that would be delivery, number 1, efficacy of the reagents, 2, and then safety, number 3. And maybe you agree with that. You were late to that discussion, I think, but that's what we all came up with. So with that one, I'm going to ask number 1, and I think I'll ask Deb to take the question first about delivery, because it seems to be the paramount question in a lot of ways, but it's going to decide which diseases we can actually tackle first based on the tissues that become most accessible to the reagents. So.
Yeah, thank you. So delivery is a question that everyone raises as one of the most important. I think one thing we have to remember is while CRISPR genome editing is somewhat new, a few years in our world it's very new, delivering genomic medicines and nucleic acids is not new. And so there have been a lot of data generated around the topic, AAV work, LNP work, et cetera. Mostly in thinking about siRNA and gene therapy, traditional gene therapy, gene augmentation work. We are really standing on the shoulders of giants there. There's a lot of work out there, and I think when we think about delivering genome editing reagents, the needs that we have are very different. So we need short exposure. We don't need long persistent exposure. We are thinking about dosing. We don't necessarily want the highest levels of exposure. So the delivery challenge is actually a lot, we have to look at it again with a new lens. So there's a lot of work out there. I don't think it's an unsolvable problem. I think it's largely what I would consider an engineering problem and just a lot of work to continue to be done. But there are a lot of amazing tools out there that we just have to systematically go through and understand what cell types we want to get to, what are the best ways to get there, at what levels of exposure and distribution, and go through all those. And I think there's going to be different delivery vehicles for different types of delivery modalities that just need to be really appropriately analyzed.
So is Editas leaving that to the scientific community to solve for them, or are they actually partnering and doing this themselves?
A little bit of everything. I think that we are exploring a lot of delivery internally, but I would say that we're not doing, there's so much work already done. We're not generating a lot of novel delivery methods internally. We're doing a lot of analysis of what's been out there, and that's both internally and with a lot of external partners.
Yes, I think for CRISPR therapeutics, so if you want to get access to any cell types in the body, some of the cell types could be a challenge. For example, even for neurons in the brain, which is a main focus in Biogen. So if you want to deliver systematically by circulation, that could be a challenge because of blood-brain barrier. But then if you find a way to inject the therapeutic reagents early on in life into the cerebrospinal fluid, in those cavities, and then there might be a way. The way the experiment in mice goes, there are some AAV subtypes. So if you inject into postnatal 1 or 2 day mouse embryos, and they can actually be taken up quite extensively by neurons. So in mice, it could be up to 70% or 80%. So I think depending on the cell types, so if you focus on some hematology-related illnesses, and you can do ex vivo approach, taking hematopoietic stem cells or CD4, CD8 cells. And so I think for CRISPR therapeutics, delivery could be an issue. But then if you choose the therapeutic indications and the cell types, then you may be able to find some low-hanging fruit there that you can start to tackle with the CRISPR.
Yeah, absolutely. I would agree with that. I would also want to say that we make the assumption that small molecule have 100% success in targeting cells, in particular in oncology, targeting all the cancer cells. But we all know that this is not the case. So I think there is an element of really understanding what is achievable with small molecule, and maybe not be too stringent of what we're expecting from CRISPR therapy. I also think that there's always been an enormous activity in the area of delivery systems, and I think we should encourage that. We should encourage completely different approaches maybe that would emerge from the work that is early but promising. And hope that this will open new opportunities for tumor indications that are not low-hanging fruit, but potentially would benefit patients.
Yeah, I think your comment about small molecules is an excellent comment. I work at Merck. We deal with small molecules, antibodies, and other biologics. And people assume small molecules are specific, but at certain concentrations, there's always off-target with any therapeutic, just as there will be with CRISPR. Perhaps it can be more specific than small molecules. And this is a Merck position, but I suspect that someday we'll kind of see some small molecules as sort of an archaic type of therapy where we're putting a compound in a body and with knowledge about off-targets. I mean, we know what the therapeutic window is for many of these, but the off-targets we can't measure as easily as we can with the genome sequence. So with the small molecule, there's just a complexity that we can't possibly measure, but with the genome sequencing or other technologies that have been talked about today, I think we have a chance of at least measuring that or estimating that with a little more specificity.
Well, it's nice to hear someone admit there's off-target activities with small molecules besides me. So facing delivery issues, a facet of that, and this now we kind of somewhat gravitate to some of the other, the second thing I pose, efficacy. Even in our own labs, when we talk about how we're going to do a gene editing project, we have to think about, well, what modality are we going to deliver? Is it going to be RNA-RNA? Is it going to be RNA-RNP or DNA or a virus? And that within itself then always is going to affect how we deliver it. And without those overarching questions as to which is the best way to do it, how do you decide therapeutically how to model a delivery system if the CRISPR reagent itself is going to change in any dramatic way?
That's a great question. Ultimately, we'd love to deliver RNP. And the idea behind that is because you actually understand stoichiometry, you have a one-to-one concentration of the therapeutic modality, it has a half-life, it's much more easy to get your hands around the exposure, the dosing, all of that, which if you're delivering DNA or RNA, and particularly viral where you have guide RNA and Cas9, being expressed under perhaps different promoters and needing to translate and become a complex, you have a lot less control over that stoichiometry. So aspirationally, I think we'd love to get to a RNP modality. Of course, the delivery vehicle or delivery method you use is limited based on the type of tissue you're trying to get at. So that's not where the technology is today. So I think the short answer is you take it in stride. You start where it's technically feasible today to get to the appropriate level of transduction to have a therapeutic impact. But you continue to work towards something where you really understand the dosing. And that is the therapeutic window, and the dosing is going to address things like safety and efficacy as well.
I have the impression that certainly with CRISPR, from a drug discovery perspective, we're a bit in a limbo, because if you think of siRNA and shRNA, for example, we had this continuum doing things in vitro, in vivo, and projecting what's going to happen in patients. And I don't think we can do that as easily with CRISPR, and that I think produce an unease. Certainly, in some of these technology and how applicable it's going to be. But I think it's good actually to be in an area where you have to invent a new system, because now scientists come back and say, well, we want to target primary cells now, or we want to target difficult to transduce cells. I think it's a great challenge, and to your point, I think we should try to really explore all the different modalities that we can apply to each one of these subjects.
Yes, I think if delivery is not an issue, ribonucleoprotein probably, I like that idea, because you can control the half-life. It has a defined half-life. But then if delivery is an issue, viral vectors, AAV, could be a choice there. Yeah.
With AAV, you have like one shot at treating the disease, right? Because they're going to have an immune response on the second application. And you're already kind of going uphill with immune responses with Cas9. Is the field really just looking at LNPs as being the solution? Is that where the real focus is? Or are you open to anything?
So that actually opens up a whole new question of how do you pick your diseases to go after, right? So I think there's a big piece around both the technical feasibility. What is the type of mutation that you're trying to address? Is it a knockout? Is it a knock-in, or replace an exon, replace a single base pair, whatever the mechanism that you're trying to get at. And I think really understanding how technically feasible that is within the delivery modality. I mean, AAV, for example, perhaps not an option if you actually need to deliver a donor DNA. So that takes a whole bunch off the table, unless you have a 2-AAV approach, which is also being explored and is possible. So there's a lot more than just, is AAV the answer? I think immunogenicity is a great, one great thing that needs to be considered, or one thing that needs to be considered, but I think there are multiple things that are considered when you select a target to go after.
So let's move on to efficacy. Because this is, again, both at the reagent level and at the therapeutic window level, how much editing can you generate, and how much editing do you really need? And again, bringing it back to a discovery perspective, where there's this field goes so quickly. Every week there's a new modality. C2 comes out last week. I mean, how are you dealing with the shelf life of reagents that may be completely obsolete within the next several months? So how do you develop a clinical development plan around a technology that is so fluid that where you start right now may be nowhere where you end up? I'm sorry, Deb.
I'm hoping. No, I think the pace that the field is going is spectacular, and I think that's just the amount of work that's being done in the number of labs that work is being done in is amazing and continually growing, and the toolbox is just getting bigger and bigger. I think there are a lot of leverageable things that you put into the toolbox, understanding specificity, understanding off targets, for example, exploring the various delivery modalities and understanding what delivery modality will work with what type of nuclease, with what type of mechanism of repair you're going for, et cetera. But I think you're right. I think that the expanding set of tools, and particularly on the nuclease side, is great, and it just gives you more opportunities to think about indications. So perhaps a PAM, identifying the right location in the gene to make a mutation, is difficult because of a PAM. Well, now you have new evolution, directed evolution to improve Cas9 for new PAMs, or you have now different Cas9s that have no PAMs, or CRISPR nucleases that have no PAMs. So I mean, you have new tools to get at a number of diseases. Again, I think the part that those are amazing engineering questions, and the work that's being done there is I said spectacular. I think identifying the right targets and the right biology to go after is actually the really difficult part. Understanding the nuance of the repair, the right model system, the assays, the biomarkers, when to intervene, the type of efficiency you need, and the right number of cells to do all that, that's actually a much harder problem to understand and make sure that you can read out in the course of the clinical path.
I think your question about technology also applies to everybody using it in the laboratory here. When do I drop my current toy and pick up the new toy because it looks more interesting and has greater potential? And I think our strategy has been largely today to stick with the Strep biology's Cas9. A lot of the systems have been worked out, the algorithms for design, delivery systems. There's still enough unknowns with that. What we found is trying to pick up some of the new reported technologies don't quite work as expected in some cases. Some of the strategies for optimizing HDR, we've seen some work from the Broad on optimizing Staph aureus Cas9, where things haven't been actually as described. And so I think that sort of conservative approach is to stick with the technology now, unless we see an obvious enabling in technology that's proven out in the literature to work in more than one set of hands. As you said, there's a new paper a week on a new technique or new strategy. And so I think having some ground truth you can work with before moving on is critical.
I mean, I'm curious. So 2 points. I think the repair question, I think, is a very good one because, in a sense, CRISPR really, in a sense, has made or rediscover some aspect of DNA damage repair, which I find kind of fascinating. Having this tool that can now interrogate a pathway that we thought we knew for, years and years. And also, I guess, this great knowledge that you can really influence, which type of modification you want to make. And I think this is quite powerful. On the, the pace of the change and how that influences therapeutic development, I mean, I think we're going to have to revisit what we call bioequivalence, I suppose. You could have very different strategies that, overall deliver very similar therapeutic benefit. And maybe, we shouldn't focus on what is the technology that allows you to drive that. It's more, well, are they equivalent in terms of benefit for the patient? And maybe that's all that matters. And safety, of course.
It's great that we have all the choices we have nowadays. CRISPR has, as a tool for genome editing, has only been around for 3 years, and I think we're all seeing the breathtaking progress in the last 3 years. But so I have been using CRISPR to engineer mouse models, and I have never seen anything that has been working so well. So when I think about for CRISPR for therapeutics, the low-hanging fruits are going to be ex vivo manipulation of cells and then transplant the cells back to the patient. And then in terms of the disease indications, it may be utilizing the NHEJ mechanism to job out an area of the genome that may have created due to the mutation an aberrant splice donor or acceptor site, or those repeat expansions. So when we think about this as the low-hanging fruit for therapeutics, and the classical version of the CRISPR actually has been working so well, meaning the Cas9 protein or the gene. So when I use that for mice, and from time to time, it's not unusual at all. You see the editing efficiency being 100%. And particularly when you're using CRISPR to engineer indel mutations and in an intron, you have all the choices of the guide, the PAM sequences, and it's possible that you choose a guide which is so efficient, that's 100%. So on the one hand, I'm happy to see the progress and all the choices, CPF1 and all the new versions of the enzyme, but then I like to think that the first version that came out and actually has been working so well.
Okay, so many of the talks today and have been focused around off targets and algorithms to predict off targets, methods to detect off targets, philosophies about if you care about them or not. George Church this morning kind of just inferred that the problem has been solved with the engineering of Cas9 to be destabilizing the non-invasive guide strand. What do you guys think? Is it solved?
You can't proven negative, so it's hard to say. You only see it when you see an off-target, right? So what we can tell you is if we do unbiased, comprehensive sequencing, we can identify guide RNAs with no measurable off-targets. But we're also at the level of sensitivity of detection. So it's ultimately, I think, going to come out to a safety study. I mean, it's a mutation in a region that is oncogene is going to be a lot different than a mutation in an intron in an area that makes no difference. So I think it's going to come down to what is the potential off-target if there is an off-target.
I think it'll be context specific too and delivery specific too on the way the material is delivered, if it's persistent, whether versus an ex vivo treatment with an RNP. I think it'll vary. I've heard lots of conjecture. There's been papers where there's been a couple of guides examined at all possible off targets. There's some whole genome sequencing data that's pretty convincing. I'm not entirely convinced of the specificity yet. I mean, I'd like to see a more robust study on a larger genome scale study on a larger set of targets to get a true confidence, or at least high confidence in one particular target. It sounds like you guys have got some pretty in-depth information. I've seen some stuff on iPSCs as well that looks pretty convincing, but I think it'll be context and delivery specific.
Yeah, I'll just add one thing. I think I think you're absolutely right. Context and delivery specific, which is, I think, a big piece of having a appropriate dosing or exposure level, right? Once you have 2 alleles in a cell, once the cut is made, you're done with that reagent, so you don't want it hanging around. But the other thing I just want to mention is it is a while back, and it was a different technology, but the Sangamo technology, the zinc finger technology, they've gone through this already. They actually did do a phase 1. They took their cell dose, they split it up amongst a bunch of mice and went in and did their safety trial. And the agency is seemingly understanding and reasonable in terms of the types of evaluation they're asking now. I'm sure that was several years ago. I'm sure they'll ask for more, and I think we're waiting to see what that is. But I do think it's an answerable question in terms of relevance and context.
So I'll just going out of turn here a little bit, but I just wanted to add. So I mean, obviously human beings tolerate off-target hits because we get bombarded with cosmic radiation all the time. I'm going to fly home tonight and get a dose of it. So it's not necessarily off-target. It's how that affects the fitness of the cell, and making it cancerous is one thing, but there are other ways the cells can be affected in their fitness. You're not going to be able to predict. I mean, what do you guys? How do you anticipate you're going to look at the fitness other than just if you've created a malignant cell?
I mean, in terms of the fitness of the cell, if you think about what could possibly happen, I think we saw with CAR T therapy, there was significant immune response to the chimeric antigen T cell therapy. I think so. There's a possibility of inducing immune or cytokine storm. I think the tolerance for off targets is going to be dictated by the particular disease you're going after. Not like oncology is typically on the forefront of some treatments, given the sort of dire nature and the unmet medical need. And so I think for some very severe diseases, the tolerance for potential off targets will be much higher than it is for other diseases, where you would require a chronic treatment. It's no different than drug discovery. For small molecules as well. I guess I'm sort of relating to, what's that?
It's permanent.
Well, yes, the CRISPR is permanent, and it's also a living system, a living treatment. But even in small molecule, we talk about covalent versus non-covalent inhibitors, where you have very tight binding. You can have persistence of the molecule in the body for months that you can pick up. And so I think those are some of the distinctions that'll be important.
One of the things I was pondering was whether we really have a good grasp of variability across patients for their repair capacity. NHEJ versus homologous recombination, and whether or not this will influence the efficacy of the editing, but also to a certain extent the spectrum of target activity that may appear. And I don't know if there's active research in this area to understand what is the variability across the population, especially in subpopulation of patients that we wish to treat. We make the assumption that this is all equal, but maybe not.
I think that's a good question. I think with CRISPR, some of us sort of had to relearn some of the things we learned with SII technology, and I think, you know, the reference genome is a reference of chimera of sorts. You know, we start dealing with cell lines, and then people bring assays to us, and all of a sudden you have a tetraploid cell line they didn't think about, but the assay works really well. So you have to deal with that. And then, of course, there's significant allelic variation individual to individual. I think there was a question earlier about maybe what kind of region would you pick, hopefully devoid of SNPs, but in some cases that's going to be unavoidable. And so you'll have to come up with strategies or take that risk as well. Maybe there'll be a personalized genome sequence available for particular treatments if that's a region of high variability.
So I'd like to open it up to the audience. Any questions like to ask the panel, please step up to the mic. If not, I want to throw out what's everyone's reaction to the NIH approving CRISPR therapy and for CAR T therapy at UPenn. I think that kind of surprised a lot of people, didn't it?
For the field, I think it's great. I think it's a really positive sign that this is an area that can continue to be explored and can move forward. Of course, it's the RAC. It still has to go through the FDA, so it's not approved. There's a lot more work to be done. And I think there are a lot of data to be generated, so I think we're all looking forward to seeing the data.
Yeah, I think the one thing I was a little surprised, there were still, we talked about off-targets and consequences of, you know, sort of impartial mutation, or they're trying to knock out essentially 3 genes, TCRA, B, and PD1, in addition to introducing a NY-ESO target. I was surprised at the sort of vagueness of the answers regarding specificity off targets, what percentage of editing was required, and so I felt like they were coming with an idea of what they wanted to do and maybe hadn't, either they were unwilling to divulge or they hadn't fully thought through all of the details, and that'll obviously have to come later when that goes in front of the FDA. Right now it's just a recommendation. I think it's going to face a lot more increased scrutiny, at least I would hope so.
No, I would agree. I think it's extremely exciting. You know, I think there'll be a lot of focus on what's happening next, and yeah, I hope it's not we're learning as we progress, but you know, maybe that's the nature of what they're trying to do.
So we only have a question?
I was going to throw it out there for the panel. We talked about the low-hanging fruit. I want to just think about the higher-hanging fruit of, will CRISPR as a therapeutic ever get to the point where you have a germline disease, say baby is sequenced at birth, known mutation, where CRISPR can be used as a technology for every cell in the body to repair that mutation? Will that ever happen, and if so, what's your over-under on the number of years until that's in clinical practice?
My science fiction is that human beings will be transgenic for Cas9 eventually. We'll just express it. We'll be tolerant to it, and then we can just inject a guide into our cells whenever we get a mutation we want to correct. That's my science fiction story.
Yeah, mine is that we have a Cas9 somewhere in our body we're going to be able to use, and we won't need all this.
I like that. I was going to say, I think, I don't know about every cell in the body. I'd say, you know, within the next 50 years, I think we'll be able to edit a substantial portion of our cells. You can imagine cures for, you know, editing T-cell receptors for antigens for peanut allergy or something more mild other than just rare diseases or hematological malignancies or things like that.
I actually think that I like it that nowadays it's so easy to sequence the entire genome and so affordable as well. So if we have a therapeutic indication of those that we can find a guide that's very efficacious, and then in conducting the safety profile research, and we can actually expose human cells in a culture dish to the guide and then sequence the genome for an unbiased assessment of the safety profile. So if a CRISPR therapeutic strategy is proven to be so efficacious as well as very safe, and if it would come to the point we need to correct a germline mutation, maybe it will happen. So.
Yeah, I would say, I mean, probably if you talk about every cell, perhaps if we get to enough point where we're confident of safety, germline, editing of the germline, you could imagine parents being screened for potential risk, and then germline editing probably a potential for an application.
So we just have another minute here, and I just want to throw out to the panel, what paper in the last 3 months you've read that really kind of shook your world a little bit, or you saw would have the greatest impact going forward on therapeutic applications of CRISPRs? Joel?
I think, so I work in a genetics department, and the goal is to use human genetic association to identify therapeutic targets. And, the biggest challenge, CRISPR is great at knocking things out, but it's still very inefficient at creating point mutations to try to recapitulate some of those genetic mutations we see in humans and cell lines and mouse models. David Liu's lab has published a number of good papers. They had a recent paper where they've sort of coupled Cas9 to a deaminase to make specific point mutations. So I think that was one that was particularly exciting. And I hope we continue to see further refinement in our ability to make very specific edits, not just sort of uncontrolled deletions relying on the cell's machinery.
So mine actually kind of goes towards what you mentioned earlier with this expanding toolbox. I think both the Argonaut and the C2C2 paper, just this expansion of what we can potentially do with this type of technology, from the perspective of choosing the types of indications we can go after and the nuances of the mutations to address some really difficult indications that have no other sources of treatment, is impressive. So the expanding toolbox, it's Argonaut, the C2C2 and David Liu's work, etc.
I like the paper from Dr. Jacob Kang's work that was published in March-ish. What they do is, of course, one thing that's always that's a challenge for the field is using CRISPR for therapeutic interventions, particularly for correcting a point mutations mediated with a donor oligonucleotide. So it's very important for us to understand how the damage is repaired. And I think their work really is taking one step further, trying to understand every step of the way what happened, how it binds to the target, how it cleaves the DNA, and then how the ends are released. And then if we understand that process, maybe we can manipulate it in a way to promote more efficacious HDR. So for that reason, I like that paper a lot.
Yeah, so I think two papers, we discussed this earlier, were published almost simultaneously by the Broad and by Novartis, who analyzed a large number of the genome-wide screens, and actually both identify that, it's potentially artifactual, but it's potentially of significance also that if you have amplified regions, the genes that are targeting genes in these particular amplified regions, because you have multiple cutting in a defined regions, that seems to induce lethality to a greater degree. And I think you can interpret that potentially as off-target activity, but it's interesting that it is in amplified regions, and it's also interesting that both teams kind of came to a very similar conclusions, in a sense, as to the significance of the findings from these genome-wide screens. So it's pleasing to see that there's enough genome-wide screen that you can actually analyze the data in this way and find these type of observations. Also interesting that there's one paper that is from industry, so we're not that far behind.
Yeah, so we're about 3 decades into gene therapy, a couple of approvals for really rare diseases, 2 decades into RNAi, still awaiting an approval. Should we be more optimistic than that your field will produce an approved drug sooner than those fields?
I don't blame you for saying that at all. So, but I would feel optimistic. I mean, I've been in genomics 20 years. I've been part of those people that have rang the bells about disruptive technologies in RNAi. I spent 10 years in the field. This technology is completely different. This technology is actually solving problems. It's not just creating new ones. I'd be very surprised if we don't see this leading to something substantial in human medicine.